Okay, we ended up here last time on iron, and as you can see that, as we go through this about iron regulation, I want you to keep in mind a couple concepts that are directly related to this, and one of them is that, remember, ferritin is one of the main iron storage proteins that we talked about. So it's a protein expressed in lots of cells in the body, including enterocytes, but other cells as well. And in general, the more iron that a cell has, the more ferritin it has. So if iron goes up, ferritin goes up. If iron in a cell goes down, ferritin levels go down. Transferrin, what's transferrin? That's the one that transports iron in the blood. Transferrin binds transferrin receptors on the cell's surface. So if a cell needs iron, it increases the number of transferrin receptors. If a cell has high iron, it can decrease receptors. So when we go through this information on iron regulation, those are important proteins to remember. Transferrin receptor and ferritin. Last time we talked about iron sulfur clusters, and we said that cells can make clusters called 4 iron 4 sulfur clusters. And proteins actually use these clusters to basically assume a specific structure so they can function in a specific way. Aconitase uses a 4 iron 4 sulfur cluster like this one here. So for example, you could say this protein would be aconitase. And we said that aconitase is a Krebs cycle enzyme. The same protein, when iron levels are low, doesn't function as aconitase because the cell has more 3 iron 4 sulfur clusters. That cluster has a different shape, and that causes this protein to function as iron response element binding protein, which means nothing so far because we haven't gone through this yet, right? So one protein, 2 functions, depending on iron levels. Now this is how iron response element binding protein can change to aconitase or vice versa. Here's a ribbon structure of a protein. And this protein, for example, it is wrapped around the 4 iron 4 sulfur cluster. So this would be an example of aconitase structure. So in high iron conditions, it would basically bind that sulfur cluster and it would look like this. And then it would function as an enzyme. In low iron conditions, this same protein now has a different structure. It instead binds this molecule. This molecule is an iron response element in mRNA. So let's look at this. So this little pink ribbon structure here is basically shown right here. This is an mRNA molecule. Here's its 5' end, 3' end. And normally when have you guys talked about secondary structure in RNA or anything? A little bit. A lot of times we think, oh, you know, and I'm guilty of this, I'll draw RNA like this. Okay, here's RNA. Single-stranded, straight line. And in actuality, in cells, it doesn't do that. It actually kind of folds on itself. One thing that we haven't talked about in here about RNA is there is a particular part on RNA that's not translated. So we call that 5' untranslated region. At the other end of the molecule, this is untranslated. So what do we call this? 3' untranslated region. And we have this as the coding region. Why do we call it the coding region? Because it's translated. So in other words, the ribosome comes here, binds, moves through the coding region, and that ribosome is translated into a protein. So this part of the mRNA is actually coding the amino acid sequence. So this ends up being part of the protein. These nucleotides in the coding region determine amino acid sequence. Now that doesn't mean that the 5' and the 3' UTRs are not important. Here's a classic example of why they're important. Some 5' UTRs, and I should write this, contain these structures. These are called stem loops. Stem loops. Got a stem, got a loop. We used to call them hairpins because they look like the old hairpins. Used to do that. Okay. This stem loop represents an iron response element. Some mRNAs have iron response elements. And if we look at this slide, this shows the nucleotide sequence. And you can see what's happening here. We know that in DNA, nucleotides can form complementary base pairs. Same thing can happen in RNA. G's bind with C's, A's bind with U. So these are basically hydrogen bonding, and these nucleotides are now forming this stem loop structure. The reason that's important is specific proteins can bind iron response elements. So this provides a protein binding site. And that's what's happening here. So what would you name that protein?
So the IRE is a protein, like a protein we're binding to that?
No, the IRE is the iron response element. Remember in 328 we said an element is a short segment of nucleotides? In this case, remember the vitamin D responsive element, metal responsive element. The iron responsive element are the nucleotides that make up this stem loop. Okay, that structure provides a binding site for a protein, and that protein is called the iron response element binding protein. Pretty creative. That's what this is showing. So if you're going to name this protein, it's IRE-BP. And this protein, Aconitase. Same protein, two structures, two functions, determined by iron status. So in high iron, we have this shape. Why? Because there's enough iron to form more 4 iron 4 sulfur clusters. In low iron, we have more 3 iron 4 sulfur clusters. So when this protein is in a cell now in low iron conditions, it's IRE-BP. Easy?
If a cell becomes, gets low iron, then does it change into the Aconitase, or does it?
It can, yeah. Can. Typically what happens is that protein doesn't stay around long enough, it's turned over, and then when a new protein's made and the iron status of the cell is higher, then more of that, kind of that novel protein or new protein functions as aconitase. Okay, now because this says IREs are present in several genes, and they're in the genes, but they're especially in the mRNA, because when that gene is transcribed, you end up with the mRNA. So be careful here, we're dealing with mRNA, not DNA. Now IREs are present in several genes and their mRNAs, including transferrin receptor and ferritin. When this paper came out years ago, this was the first, what they called, kind of molecular switch for nutritional regulation of protein synthesis. And these guys, the guy that did this was named Richard Klausner. He was an NIH guy. I think he's an MD, PhD. His group got the Nobel Prize for this. So this is this is a classic diagram. It's not perfect, but I'd rather use this one than something that's been modified. This is iron regulation of transferrin receptor and ferritin. We have 4 different parts to this. My suggestion to you would be learn each part individually, learn how it works so you don't have to memorize this. If you memorize this, it's going to drive you a little nutty, maybe. Here's iron deficiency, top panels. Iron overload or high iron is down here. Let's look at this one first. In iron deficiency, this protein would be functioning as, which one? IRE-BP. In other words, if iron response elements are present, this protein will bind to it. Here is the ferritin mRNA. 5' in, 3' in, coding region. Ferritin mRNA has an IRE in the 5' UTR. So in this scenario, what they're showing is, see our IRP? They abbreviated iron response element binding protein as IRP, so it's the same thing. In this case, IRE-BP is binding to the IRE. Normally what happens during translation is a ribosome would bind here and move through the coding region. If IRE-BP is bound to the IRE, translation is blocked. Make sense? I'll ask you this way. Who gets that? Okay, I'm not going to ask who doesn't. I'm assuming the other people hasn't clicked yet. So normally what happens in the cytosol, if this was an mRNA molecule, a ribosome would bind here, right? We normally do this. We say, okay, here's mRNA. Ribosome binds, moves through the mRNA, makes a protein. Protein comes out. If IRE-BP is bound to the IRE in the 5' UTR of the ferritin mRNA, translation is blocked. The ribosome cannot bind and translate the mRNA. So what would happen to the protein level? Up or down? Down. So let's look at the context. In iron deficiency and low iron, do cells need to make more or less ferritin protein? Less. So in iron deficiency, less ferritin protein is made. Remember that last part that I said? You can make the other part work. In iron deficiency, less ferritin protein is made. And that makes sense, right? Don't need it. Let's look at this one. Let's say, for example, a person has high levels of iron in cells. In high iron, there's more aconitase, less IRE-BP. So the protein, this is showing higher iron and bound to IRE-BP. So if you want in your notes, see where it says IRP, I think a better way to label that would be what? That's aconitase. They have iron here that's supposed to represent that 4 iron, 4 sulfur cluster. Got it? So now you have a protein that's aconitase. It's not binding mRNA. It's not looking for an iron response element binding it. So in other words, translation is not blocked. So in high iron, aconitase doesn't bind the IRE of ferritin mRNA. So we have active translation. And does that make sense? In high iron, do you want to make more or less ferritin? More. So think about the enterocyte. If a person has consistently high amounts of iron in those enterocytes, more ferritin is going to be made. That allows those enterocytes to bind some of the iron and decrease its absorption. Got it? Okay, let's look at the other two. This is so easy. 7:30 in the morning. Here we go. Transferrin receptor, iron deficiency. So let's take a step back and say, in iron deficiency, let's say a cell has low iron. What do you want to make, more or less transferrin receptors? More. Same proteins involved, aconitase, IRE-BP. The difference in regulation in this case is the position of the IREs. In the ferritin mRNA, they're 5'. Look where they are in transferrin receptor. Be careful, this is receptor, not transferrin. In the receptor mRNA, they're 3'. That means the effects are opposite. And here's how. And we haven't talked about this. I don't know if you can see this. This is a very accurate three-dimensional representation of an enzyme called 5' to 3' RNase. Have you ever heard that term? What do you think it does? It's an enzyme that degrades RNA. Normally in cells, RNA doesn't stay around for long periods of time. One of the reasons is we have what are called 3' to 5' RNases. They degrade mRNA. And that allows basically translation to be regulated in a dynamic fashion. We make RNA, it's degraded. In order for us to continue to make RNA, we have to have a consistent stimulus. If we don't, what that means is translation levels are altered based on certain stimuli. Okay, so this is kind of, the guy that figured this out was, this is called mRNA decay and mRNA half-life. The guy that figured this out, this wasn't that long ago that they figured this out. Remember the poly A tail? Remember that from molecular biology class? Or biology? What's the RNA, what do they say that poly A tail is for? They say the longer that poly A tail, the more that RNA stays around. The longer it does. That's one of the functions of that poly A tail. Why? Because these enzymes are degrading from the 3' end. So if you put a bunch of tail out here, it takes longer for this mRNA to be degraded. Okay? That is unless mRNA is protected. In iron deficiency, the bifunctional protein functions as IRE-BP. It binds the iron response elements in the 3' end of transferrin receptor mRNA and protects it from RNase degradation. So when IRE-BP binds IREs in the 3' UTR, it protects the mRNA from RNase degradation. That's why it says stable mRNA here. So theoretically, if an mRNA is around longer, can more of it be translated? So that typically means translation stays high. So in iron deficiency, should transferrin receptor levels go up? They should, and they do because of this. The transferrin receptor mRNA is protected from degradation, and that allows more translation to occur. See the difference? There's no IRE here, so translation's not being inhibited. So ribosomes can bind and translate as long as the mRNA is there. And in this case, more of it's there because it's being protected. So again, all you got to remember, big picture, is that iron deficiency transferrin receptor protein levels go up. Got to remember that. So if you want, make a little box. Make a 2x2. And your top right corner has, in iron deficiency, transferrin receptor levels go up. Let's look at the other two, because that's so easy. Let's go one more level. Level 3. This is high iron. So we would expect this protein to be Aconitase. It's not binding mRNA. So in high levels of iron, we have active translation of the ferritin mRNA. IRE-BP is not binding here. It's not blocking translation. So we have active translation of ferritin mRNA. So in high iron, would you expect ferritin levels to go up? You would, and they do. So this square means in high iron, more ferritin protein is made. Over here. Now we're talking about transferrin receptor. If a cell has high iron, should it make more or less transferrin receptors? Less. So in that case, the IREs are not bound by IRE-BP, so we have an unstable mRNA. In other words, it can be degraded by RNases. So in high iron, transferrin receptor levels go down. Because the RNA is more quickly degraded. Make sense? You might have to look at that once or twice more. Take it step by step. You got 4 options here. The level of detail that you should know this is, I can ask you basically to reproduce this, and I won't have you do them all at once, but basically I might say, tell me why in iron deficiency ferritin protein levels are increased or decreased. Tell me the mechanisms, include mRNA, include IREs, that level.
This may be like too complicated or too much detail, but if the RNase moves 5' to 3'.
3' to 5'. Okay, so this is, keep this in mind. I might ask you, based on the known fact, let me put it like this. What's DMT1? This is a long time ago. This is Tuesday. DMT1. Divalent mineral transporter 1. It's on the brush border membrane. And I said at that point, one of the points of regulation of iron absorption is DMT1. I said in iron deficiency, DMT1 levels go up. When a person has high iron status, DMT1 levels go down. DMT1 mRNA contains, yes, IREs. Explain to me how the IREs regulate DMT1 levels in iron deficiency and high iron. So what's one of the things you have to do to sort that out? Where are the IREs? That's what you got to figure out. So go through this model, figure out where the IREs should be on DMT1. That's where they are, and then explain that.
Will you say that question?
Explain how iron levels in a cell regulate DMT1 protein. Include IREs. So in low iron, DMT1 should go up or down? Should go up. So which one of these does that more closely resemble? And then you got it figured out. Okay? There's a few more of those, but that's, I think, a great example of that. Okay? Now this is a slide that your textbook has that is, it's kind of a nice overview of what we just talked about. We said, for example, in high intracellular iron, we have 4 iron, 4 sulfur clusters. So this is IRE-BP. This is showing it's part of the TCA cycle. And here's Aconitase. So this actually, a better name for this one would be what? Aconitase. Functions as Aconitase. And in the Krebs cycle, that enzyme converts citrate to isocitrate. So low iron, Krebs cycle activity can go down because this protein is now IRE-BP. It's not functioning as Aconitase. Now when they first did these experiments years ago, they pulled out this protein that was binding to mRNA, and they did some pretty elaborate cross-linking experiments. They kept coming up with this protein, they isolated it and they sequenced it, and they kept coming up with a Aconitase sequence. And they kept thinking, we've somehow contaminated our protein purification with Aconitase. They repeated it over and over again and finally realized it's the same protein. So in low iron it does this. Now this isn't specific enough because it doesn't show the location of the IREs. But overall it shows, in this case, in low iron we have decreased translation of ferritin. And in low iron we have increased translation of transferrin receptors. But you need to be able to tell me where the IREs are in each of those. Okay? So let's take a little bit bigger picture of functions of iron. What else does it do? Well, it functions as part of proteins. Right? Some proteins use heme. Some proteins use iron sulfur clusters. For example, we have hemoglobin and myoglobin that use heme. Cytochromes use heme. ETC enzymes, for example, move single electrons down the chain by oxidizing and reduce the iron. In ETC, we have both. Some use iron sulfur clusters, some use heme. So ETC is also iron dependent. And if you ever do a mitochondrial isolation, that mitochondrial prep is usually like a rusty brown, and the reason is because of all the iron in there that's in Krebs and ETC enzymes. It's also important for cytochrome P450 proteins, which are ER proteins involved in the detoxification of drugs and alcohol. So it's kind of a protective pathway that depends on iron as well. Then of course we have the Aconitase IRE-BP example, and there's lots of other proteins that actually require iron. Another example would be like the proline, lysine hydroxylases we talked about in vitamin C. So we know there's a lot of requirements for iron, but on the other hand, we talked about this. Iron can be toxic if it's in higher levels. We're going to skip the Fenton reaction. Because of iron's toxicity, there is a UL. The UL for iron is 45 milligrams a day. The UL is 45 milligrams a day. Is that a lot relative to the, what did we say the DRI is? It's an RDA. What was it? Eight and 18 for men and women. So 45, I'd say that's not that high for UL relative to its recommended intake. And the reason it can be toxic is because it can become, or it can function as a pro-oxidant. Its toxicity is because it can function as a pro-oxidant. So it oxidizes and damages other molecules if there's too much iron. It's still one of the most common molecules involved in accidental overdose as far as toxicity. So accidental iron overload is basically when someone takes excess iron supplements. It's not going to happen with food, but with supplements. So what's a population at risk? Kids. It's unfortunately too common where kids will get their hands on vitamin mineral supplements for adults, and especially the way they make them now. Ooh, I like those because they're gummies, right? And they'll consume those. Sometimes they'll eat the whole bottle, and it can cause iron toxicity. Sometimes it's so bad it's lethal in children, because if you think about it, a lot of iron in a small individual, one of the reasons it's so toxic, it can cause liver dysfunction and eventually liver failure if that iron is too high. So people need to be really careful with those supplements if kids are around. Hemochromatosis. Hemochromatosis is a genetic iron overload. And I have this in my notes, so I think it's probably right. This is the most common genetic disorder in the U.S. So it's a genetic iron overload. How common is it? Most common genetic disorder in the U.S. affects at least 1.5 million people in the U.S. What causes it? Hemochromatosis is caused by a defect in the HFE gene. So the protein that normally comes from that gene is required for hepcidin synthesis. Remember what hepcidin does? Hepcidin's a hormone made by the liver, regulates iron absorption. When iron levels in the liver go up, hepcidin's released. Hepcidin travels through the blood, causes ferroportin to be degraded. So high hepcidin means decreased absorption of iron, normally. You can go back through your notes and look at that. The problem is, in this case, there are fairly common genetic abnormalities where the HFE gene product is compromised. So what happens in this case, we don't have that protein that is now helping make hepcidin. So in that case, even when iron goes up, what happens to iron absorption efficiency? Nothing. It's not downregulated. So in hemochromatosis, the lack of the HFE gene product you could say that compromises the ability to decrease iron absorption. So in hemochromatosis individuals, iron absorption efficiency tends to be 2 to 3 times normal. And they continue to accumulate iron, even when it's at high levels. So it's continuously deposited in tissues, including the liver, eventually causing liver damage that's potentially lethal. Right, and it's really common compared to genetic disorders. So quite a few people have to get treated. Typical treatments for this, what would you do? Decrease iron intake. Usually includes chelator therapy. Have you ever heard that term chelator? We've probably heard that. Oh, chelation therapy, stuff like that. Chelators are organic molecules that bind minerals. The problem with chelators, a lot of them aren't very specific. If they bind one metal, they'll also bind similar metals. In this case, though, what's the name of that chelator? I think it's called phenanthroline is one of them, and the other one that's more common, I think it's called desferoxamine. It has a negatively charged component that'll actually bind the iron. So they're taken orally consistently, and the chelators tend to increase urinary excretion of iron. They'll bind the iron so it doesn't go into cells, and it's excreted in the urine. Third thing, what's something else we could do to remove iron from a person?
Blood.
Yeah, blood removal. Consistent phlebotomy. Initially, levels are fairly high, they'll remove a few hundred mils at a time. When iron levels eventually kind of dissipate and go down, the blood removal is less consistent, less volumes removed. But they're continuously removing blood. That occurs for the rest of their life typically. Okay? Opposite. Iron deficiency. I gave you numbers for last time about how common it is, right? Most common nutrient deficiency in the world. And it's more common in developing countries, but it's also relatively common in the U.S. Developing countries, almost half of preschool children and pregnant women have iron deficiency anemia. In developing countries, almost half of preschool children and pregnant women have iron deficiency anemia. Significantly less in the U.S. I won't give you numbers for that. It's probably about maybe 10% of toddlers, adolescent girls, and women of childbearing age. So in the U.S. it's a lot less common, but relative to other nutrient deficiencies, it is common. Iron intake is frequently inadequate in 4 groups: infants and children. And this is because in those cases, you know, they have high iron demands. So in infants and young children, this is 6 months to 4 years of age, they're more likely to be iron deficient because low iron content of milk. So this is because of low iron content in milk. Rapid growth rate. And insufficient stores. Insufficient stores or reserves. Adolescents are more susceptible to iron deficiency because of the rapid growth rate, including blood synthesis. Women in childbearing age, because of menstrual losses. Pregnant women, because of expanding blood volume. Demands of the fetus. And blood losses at childbirth. Okay, one of the things I mentioned before was a lot of people are iron deficient but don't have anemia. So anemia is the last stage of iron deficiency. So we're going to go through and kind of look at stages, and this is kind of related to your lab report as well. So we're going to go through this. This is the same table that's in your lab handout. There's a lot of information in here, so we're going to break this down into pieces, and it kind of goes left to right. See where it says normal? This next column right here, we're going to call that stage 1 of deficiency. So if you want, you can put a Roman numeral up there. So here's stage 1 of iron deficiency. Stage 2, 3, and 4. So I'm going to give you an indicator in each one of those stages. Stage 1. This is where the iron levels in tissue start to decline. A good indicator of this is plasma ferritin goes down. So stage 1 indicator, Plasma ferritin goes down. Does this seem weird? Where'd we say ferritin is? It's in cells. Some ferritin actually leaves cells and goes into the blood. More of it's in cells, but some of it's in the blood. It tends to be proportional. So if it goes down in the blood, that means it's down in cells. And why is it down in the cells? Because of that mechanism we talked about before. Low iron causes decreased ferritin production. Stage 2, this is where transport iron declines. Good indicator of stage 2. Oh, we can kind of look at this. Oops. See our plasma ferritin, normally 100, goes down below 25. That's our indicator there. Next stage, stage 2, we're going to say serum transferrin receptors go up. Normal, elevate a little bit. In this case, they go up. Remember, transferrin receptors are on cells. Some of them leave the cell surface or end up in the blood. You can actually quantify that. So they go up. Stage 3. This is what you guys measured. Plasma iron goes down. Transferrin saturation goes down. So you have more open binding sites on transferrin, so it's less saturated with iron. Stage 4, anemia. Red blood cells are microcytic hyperchromic. So hematocrit goes down, hemoglobin goes down. I think that's it. Questions on iron? Okay. Should I stop the recording there or keep going? Okay. Assuming we're still going. I think we are. Okay, let's do vitamin D. Start it. So what are some functions of vitamin D? When you think of vitamin D, what do you think of? Calcium usually, right? So one of the main functions of vitamin D is calcium metabolism. What kind of molecule is vitamin D? We call it vitamin D. It's fat soluble. Is it a vitamin? What's a better probably definition? It's really a hormone. Right? We said a hormone can be made in one part of the body, elicit response in distal tissues or cells. Made in one part of the body, causes a response in other tissues. That's what vitamin D can do. Do we also get vitamin D in our diet? Yep. Do we need to consume vitamin D? That is debatable. Okay, it depends. Most people, absolutely. So it is considered an essential nutrient. But we tend to make a lot in our body, typically. It's not what's on my screen. Okay, so vitamin D has a few different forms. What are some food sources of vitamin D? What are some really good food sources? There aren't many. Right? We tend to get quite a bit from dairy products. Why? It's fortified. Right? So good sources of vitamin D are not that easy to find. One of the classic ones is oily fish. We eat a lot of fish in this country. Overall, probably not enough. So a lot of people struggle to get enough vitamin D from the diet. Now, can they get enough from sunlight? It depends. So we're going to talk about that. We can make some vitamin D in our body. We'll go through that process. But once it's made, it has to be activated. So there's several steps along the way between diet or synthesis of vitamin D in the body that have to take place in order for that vitamin D to be active in our body. So it has some metabolic features that kind of help protect us from high amounts of vitamin D, but it also complicates the issue, for example, when we get older or we're not out in the sun, or if we have any kind of kidney or liver dysfunction, it can decrease the ability of vitamin D to be activated, and that can cause downstream effects. Okay, so vitamin D, two of the structures are shown here. Vitamin D is actually a group of vitamins. That means there's several different forms of the vitamin. Oops. Have you heard of these, vitamin D2 and D3? What's the difference? Which one's better? D3. Especially some recent studies have indicated, yeah, it's probably better to take in vitamin D3. Which one do we make in our body? D3. That should be a pretty good indicator, right? Oh, that's the one we make in our body. That one's probably better to get in our diet as well. But that debate went on for years. A few years ago, it was probably only 5 or 10 years ago, most of the supplements were D2. Now most of them are D3. They're pretty similar. Here's D2 structure. It's called ergocalciferol. Here's D3. It's called cholecalciferol. So if you need to, practice spelling those. Look at the difference. Can you see any differences? There's D2. Yeah, up here, double bond here. Not one here. And where else? Oh, here? Yeah, so we have a little bit different side chain, double bond. There's also another double bond somewhere. I thought there was. I think these are right. Yeah, so just a unsaturation here, slightly different carbon chain. Pretty similar molecules. Both of these can function in the body. So both D2 and D3 forms have what we call vitamin D activity. We're going to spend a few minutes on this one. This is the synthesis of vitamin D2 and D3. This top part, if you want, you can circle this because this one's kind of separate. So here is vitamin D2, ergocalciferol. It's made from ergosterol, a steroid in plants, and yeast. Right, plants don't have cholesterol, they have ergosterol. You've probably heard that before. Looks like cholesterol though, doesn't it? Here's cholesterol. It's pretty similar. Slight differences in the structure. In this case, we have a double bond here. We don't in cholesterol. And then slight difference in the structure here, double bond, no double bond. So pretty similar. Ergosterol and cholesterol are very similar. Plants can convert that to vitamin D2 when they're exposed to irradiation. You ever seen that? Like mushrooms are a good source of vitamin D. They are if they're irradiated. They actually expose those mushrooms to UV light, and it converts a lot of their ergosterol to vitamin D2. Then we can ingest that. Vitamin D2 goes into our cells, and we can actually activate it like we activate vitamin D3. Okay, so this is plants. This is how animals synthesize it. So you can put down here D3 synthesis are these steps. So let me tell you this as kind of not really a side story, but a reasoning behind this. A few years ago, there was a researcher. He was an MD, MPH. His name is Michael Hollick. He was at Harvard University, and he was looking at epidemiological data and basically said vitamin D is related to cancer. Certain types of cancer are affected by vitamin D status. Have you heard that? So vitamin D, we know, is involved in calcium, but vitamin D also is involved in differentiation of lots of cell types. Vitamin D tends to protect us from certain types of cancer. So Michael Hollick basically started saying, you know what, people need to get out in the sun more because it'll protect them from cancer. Sounds counterintuitive. It was so controversial when he was saying that. He was going around the country, going around the world saying, look at these data. We are decreasing our sun exposure and cancer levels are going up in these areas. Harvard said, yeah, you got to stop saying that because you're promoting people going out in the sun and increasing the risk for skin cancer. And he said, no, I'm looking at the data, that's right. And Harvard said, stop saying that. And he said, no, that's the data. And they said, you're done here. So he had to leave Harvard, and then he went to Boston. So he's at Boston University, the medical center there, continued on his spiel. Turns out he was right. Vitamin D status, if it becomes compromised, can increase the risk of certain types of cancer. So some sun exposure is okay, but we want to be careful about too much. This is the reason right here of why that kind of is true, because here's cholesterol. We can actually make vitamin D3 from cholesterol. I think as we've kind of indicated several times, our body can make lots of cholesterol. That cholesterol travels through the tissues, including it will go into skin cells. Right? In the skin, and this is actually in the sebaceous glands of the skin, basically the glands that make that waxy substance that kind of gives our skin kind of a protective layer of lipids, cholesterol is converted to 7-dehydrocholesterol. So simple conversion here. You don't have to remember structures, but you got to remember the names of these. So cholesterol converts to 7-dehydrocholesterol. When that happens in the skin and 7-dehydrocholesterol is exposed to UV light, it converts to pre-vitamin D3. So you need that UV exposure. So in general, the longer you're in the sun, the more vitamin D is synthesized.
Oh, I was going to ask, is there a certain amount of time?
Yeah, we'll go through that, yeah. Yep. Okay, so now, once you have vitamin D3, that's called pre-calciferol. So this is kind of a pre-vitamin D3. It's not the active vitamin yet. Now, there are a few options for this molecule. Continuous exposure to UV converts some pre-vitamin D3 to these molecules. And your textbook, I think, is wrong here. So we cross this out. Pre-vitamin D3 can become lumisterol and tachysterol. Two molecules that have no vitamin D activity. And I'll tell you why they're important in a second. once pre-vitamin D3 is synthesized, when it's exposed to body heat, and this is anywhere from hours to a few days, so this is body heat, basically body temperature. Pre-vitamin D3 is converted to vitamin D3, and that's called cholecalciferol. And if you look at what's happened, when that 7-dehydro was exposed to UV, look what happened. This ring opened. So the molecule opens up. Continuous exposure to heat causes this molecule to kind of flip. So it changes its structure here. See the way this structure is different here? This structure and this flexibility link between these rings allows vitamin D3 to interact with proteins. Right? So the flexible structure of vitamin D allows it to interact with specific proteins. And we'll see later on that's what's required for it to be active. Now, what do you think? What have you heard about vitamin D? Is it potentially toxic? It's one of the most potentially toxic vitamins, if not the most. So there is a UL for vitamin D. So too much vitamin D can have harmful effects. So we want an optimal level, but no more. And that's what's really controversial right now about vitamin D, is how much is enough and how much is too much. And I think so far both of those are being handled conservatively. Wouldn't surprise me for a very short period of time the RDAs change again, the DRIs do, including the UL. I think the UL is probably too low at this point.
So from this model, then there would be like a sweet spot for how long you'd want to be in the sun, because if you're in it too long, then that free vitamin D degrades.
Yeah, but in some ways that's okay, because this is what happens. Let's say, for example, I just said that too much vitamin D in our tissues can be toxic. What if you went to Avila and you stayed out there all day? Are you going to get vitamin D toxicity? Why not? Because of these molecules right here. Right? These are protective pathways. So continued exposure to UV converts pre-vitamin D3 to lumisterol and testosterol. Those are molecules that basically just stay in the skin or eventually degraded. They don't become vitamin D. So there's no chance of vitamin D toxicity from getting too much sun.
Will some of it, if you're in the sun all day long, still convert?
Yeah, so here's right. So let's say I could measure vitamin D in the blood. And let's say I started this period of, oh, it's summertime. I'm going to start going to the beach more. I'm going to be outside more. Will vitamin D levels in the blood go up? Yep, they tend to. Why? UV exposure, you're making more. Okay? But with that said, you tend to you don't make too much to become toxic levels. So it is UV dependent. And that does affect the amount of vitamin D in the blood, but that it does not reach toxic levels because of UV exposure. Have you ever heard this? Like if you live in certain parts of the country or certain parts of the world, your vitamin D levels are affected. So in general, for example, vitamin D levels in the blood in the wintertime on the central coast of California are higher than those people that live in Alaska or Canada or even Washington and Oregon in their summers, because it's seasonal. So you can actually track that. Right? If you've lived in the Pacific Northwest or anything like that, certain times of the year, it is actually linked to certain types of depression. And part of that is believed, it might have to do with vitamin D levels. So vitamin D levels go up and down depending on sun exposure in most people. We'll talk more about that coming up. But now what happens is once you have this vitamin D3 let's say it was just synthesized through this, vitamin D3 binds to this protein, and this is called vitamin D binding protein. So vitamin D binding protein is the main vitamin D transporter in the blood. Vitamin D binding protein is the main vitamin D transporter in the blood. It has high affinity for vitamin D, but not pre-vitamin D3, lumisterol, or tachysterol. So vitamin D binding protein does not bind pre-vitamin D, lumisterol, or tachysterol. So it doesn't leave the skin. These molecules don't leave the skin. Okay? So once that vitamin D, this vitamin D3, binds vitamin D binding protein, it starts circulating through the blood. That's how it gets to other tissues. Now, down here, this is an important point. Vitamin D2 and D3 have to be hydroxylated to become active. We'll talk more about that coming up.
Is vitamin D2 binded?
Yes. D2 and D3 bind DBP. Okay, so we know we can get vitamin D2 from our diet. Do we synthesize vitamin D2? We don't. So D2 is just from your diet. Where's D3 from? Diet and our own synthesis. So D3 can come from diet or our own synthesis. Let's do one more slide, then we'll stop, because the next one's, then it'll get a little bit sequential in detail. So here we go. Here's absorption and transport. Vitamin D2 and D3 from the diet is absorbed from micelles, right? It's fat soluble, goes into the gut, becomes part of mixed micelles, and then it's absorbed into enterocytes by passive diffusion. So it diffuses down its gradient into enterocytes. Overall, about half of the dietary cholecalciferol, I should say this. I'd say half of dietary cholecalciferol and ergocalciferol, so D2 and D3 is absorbed. There's no absorption differences between D2 and D3. Sorry about that. Now, in the intestinal cells, most of the vitamin is incorporated into chylomicrons. Right, so it goes mixed micelles, into the enterocyte, into chylomicrons. Let's skip to the last one. The vitamin D that is in chylomicrons is delivered to the liver in chylomicron remnants. So most of the vitamin D that is in chylomicrons, as chylomicrons go through the blood, those chylomicrons tend to hold on to the vitamin D, so it ends up in the liver. So most dietary vitamin D goes to the liver first. The D3 that we make in the skin, when it binds to vitamin D binding protein, it can be delivered to extrahepatic tissues, especially muscle and adipose. So that's a little bit different. So it's more likely that vitamin D binding protein delivers D3 to tissues outside the liver, whereas most of our dietary vitamin D ends up in the liver. Okay? Sure. Most vitamin D from the diet is delivered to the liver by chylomicron remnants. The vitamin D3 that's made in the skin is likely to be transported to muscle and adipose. Okay, at least initially. Now, the next steps that we're going to talk about has to do with modifying vitamin D so that it's active, and that has to do with hydroxylation. There's a few steps involved there. So we have vitamin D in the system, but it's not activated yet. So either from the skin or the diet, our liver and kidneys have to hydroxylate it for it to become active. Remember that from 210? Have you guys talked about that at all? Okay, new stuff then. Okay, let's stop there. I'll see you guys all tomorrow, right?